Welcome, everybody, to our gene delivery seminar series for July. I'm very excited about today's talk. We have Dr. Francesco Andreata. Dr. Francesco Andreata is an assistant professor in the Faculty of Medicine at San Raffaele University in Milan, where he leads a research team studying how dysfunctional immune responses in tissues can be reprogrammed to combat chronic infections and cancer. He earned his PhD in immunology with honors from the University of Paris and then joined Professor Matteo Lancon's lab at San Raffaele Hospital, where he uncovered how hepatitis B infection drives a distinct dysfunctional CD8 T-cell program in the liver and identified a Kupffer cell subset that can reinvigorate these T cells via interleukin 2. This work has led to key publications and initiated collaborations with pharmaceutical companies for the preclinical development of immunomodulatory therapies. Francesco's research spans chronic viral infection, cancer immunology, and liver immunobiology, examining how tissue architectures shape immune cell function. We are very excited about today's talk, and it is my pleasure to welcome Dr. Francesco to our seminar today.
Thank you very much. Good afternoon, everyone. First of all, thank you for the kind invitation and for the kind words. It's a pleasure to be here today. So as you were saying, our lab is interested in understanding how tissue environment shape immune cell function. And today, in this talk, I will focus on one very particular organ, which is the liver, because unlike other tissue, it has evolved to promote immune tolerance while remaining capable of mounting, in some cases, protective immune responses. In this seminar, I will discuss how antigen recognition within the liver determines the fate of CD8 T cells and how this process differs from classical T cell exhaustion, and how those mechanisms may help us to design better immunotherapies. Okay, the liver is probably one of the most immunological unique organ within the body because of its unique features, unique anatomical and hemodynamical features that make immune response with this organ basically distinct from those occurring in other tissue. First of all, I want just to mention that it receives a dual blood supply, approximately 80% from the portal vein, from the gut, and 20% from the systemic circulation from the hepatic artery, which result in a low pressure, slow flow hemodynamic environment, which allow CD8 T cells or naive T cells to interact with the endothelial cells without the need of all the molecules and adhesion molecules that are usually required in other districts to roll and to adhere to endothelial cells. Secondly, the liver endothelial cells of the sinusoid, which is the liver sinusoid endothelial cells, are the unique, the only mammal endothelial cells which combine two very interesting features. The one is the presence of fenestration, which are holes of about 20 to 200 nanometers in diameter, with the absence of a fully formed basal membrane. So the combination of all these features makes something which is not possible in other tissue, which is the fact that intravascular T cells, like CD8 T cells, can interact with underlying hepatocytes and scan MHC class I complexes without the need of transmigration. Of course, all of these characteristics profoundly impact the priming and maintenance of adaptive immune responses in the liver. And this is maybe most evident in chronic HBV infection, which are used in our laboratory as a prototypical hepatotropic infectious agents. But of course, it's also very relevant medical problem because despite the presence of a preventive vaccine, more than 250 million of people are chronically infected worldwide, and millions sadly die each year for complication, such as cirrhosis and hepatocellular carcinoma, which is the primary tumor liver. So there's no definitive cure. And what is very important about this virus, it's that it's hepatotropic, which means that infect exclusively the parasites in a non-cytopathic way. And the other peculiarity of HBV, which makes interesting from immunological point of view is that the way it replicates and its life cycle, it's done in a such way that is basically invisible from the innate immune system detection machinery. And so that the adaptive immune cells, especially CD8 T cells, are very crucial for the eradication of the virus. And a problem in differentiating proficient CD8 T cell and antiviral T cells, it's one of the leading cause of chronicization of the disease. So in the past, our lab has shown that naive T cells, which are normally excluded from interacting with epithelial cells in non-secondary lymphoid organs, can be primed within the liver by extending cytoplasmic protrusion. And this it's visualized here in this example correlative confocal and transmission electron tomography. If we do a 3D reconstruction and we tilt these images, we can appreciate the CD8 T cells in red, extending a cytoplasmic protrusion, bridging the sinusoidal wall at the level of this fenestration here in green, contacting underlying hepatocytes. So our previous work showed that hepatocellular priming, meaning that the priming of naive CD8 T cells directly from the circulation within this process, leads to the local activation and expansion of antigen-specific CD8 T cells. However, these cells fail to develop and differentiate into functional antiviral effector, and instead acquire a dysfunctional phenotype, which is interesting because we have done some characterization. It does not really overlap with the exhaustion phenotype, which can be found in other chronic infections or in cancer setting. So at that point, and this is the first part of the work, I would like to present a work in which we asked a key question like, what are the co-signaling receptors that govern the fate of hepatocyte-primed CD8 T cells? What is the differentiation trajectory, and how do these cells follow over time? Before going into the details, the experimental details, just want to spend an extra slide showing the model that we are using, because HBV does not infect, it does infect only human and chimps, so we make advantage of a different transgenic mouse model. One of them is an HBV transgenic mouse in which the entire genome of the HBV is knocked into the mouse chromosome, and in this model, the parasites are able to replicate non-cytopathically very high titers of the virus. The second one, which is a complete artificial control that we use to benchmark the intrahepatic CD8 T cell activation, in which we are able to transduce professional antigen-presenting cells, like dendritic cells, Kupffer cells, with the replication-deficient recombinant LCMV platform, which is driving the expression of the core protein, the core antigen of the HBV in Kupffer cell and dendritic cells. So these two different models serve as recipients for adoptively transferred CD8 T cells that we collect from the spleen of this donor transgenic mouse, TCR transgenic, in which 100% of CD8 T cells express a transgenic TCR recognizing the immunodominant peptide of the core protein of the virus. In other words, when we transfer those T cells, CD8 T cells, naive CD8 T cells, in the two models, in the first time, it gets primed on hepatocytes, and for this reason, we call, for sake of clarity, this model HC priming, so hepatocellular priming. In the second model, since those T cells get primed on professional antigen-presenting cells, especially Kupffer cells, we call this model KC priming or Kupffer cell priming. So, to address all of these questions, we started performing a comprehensive transcriptional and phenotypic characterization of hepatocellular prime HBV-specific CD8 T cells, combining single-cell RNA sequencing with high-dimensional flow cytometry, in order to obtain a unbiased view of the co-signaling receptor landscape and repertoire during intrahepatic priming. So we found that among the co-inhibitory receptor, PD-1, LAG-3, CTLA-4 were the mostly upregulated, while among the co-stimulatory receptor, ICOS-4, maybe NOX40, were the one that were mostly upregulated. So to determine whether these receptors were functionally relevant for the differentiation trajectory of those T cells, we move forward from descriptive profiling to functional reprogramming. And to do that, we treat mice with either blocking antibodies against the major co-inhibitory receptor, like PD-1, LAG-3, CTLA-4, and in other experiment with agonist antibody activating the co-stimulatory receptor. So we adopted transfer naive CD8 T cells in the HBV transgenic recipient at day 0, and 24 hours later, we challenged those mice with those monoclonal antibody, and we followed their activation over time. So I'm guiding you through those graphs. So at day 5, we saw that the co-inhibitory receptor blockade did not reinvigorate those T cells. So in the first graph, we are seeing total absolute number of antigen-specific CD8 T cells that we can recover from the liver of those mice. So here the control is in blue, and as you can see here, there were no increased proliferation if we block those three inhibitory receptor. And if we challenge those CD8 T cells that we recover from the liver with the cognate peptides, we did not observe any increase of interferon gamma production. And we did not observe as well an increase in transaminases in ALT in the plasma. On the contrary, when we injected the day after the adopted transfer of T cells antibody agonist for ICOS, 4B, and OX40, we did not observe any reinvigoration with anti-ICOS agonist, but we did observe actually the reinvigoration in terms of expansion and capacity to produce interferon gamma upon cognate peptide stimulation in mice that have been challenged with OX40 and 4BB agonist antibody. As you can see here, those T cells differentiate in effector cells, and they can kill infected hepatocytes, and those infected hepatocytes released in the systemic circulation transaminase that can be followed longitudinally in those mice. So far, we have shown basically that in acute setting, inhibitory receptor seems to be not involved in the reinvigoration of CD8 T cell activation, while 4-MBX40 agonism can actually successfully revive their differentiation trajectory. But then in chronic infection, antigen persists for prolonged periods, and this raises an important question. What happens to these dysfunctional T cells over time during chronic antigen stimulation? In particular, do they retain some dysfunctional phenotype? Are they homogeneous population, or they start to be, there's some heterogeneity? And at the end, do they retain or shape their responsiveness to this co-stimulatory receptor modulation? To do that, we set up a new set of experiments in which we actually challenge wild-type mice with this recombinant SMV core platform, so that professional antigen-presenting cells start to present the antigen, or we take the HBV transgenic mice, and we adoptively transfer CD8 T cells, and we wait for 28 days. So we were surprised actually to see that in our dysfunctional phenotype here in red, the HC prime, we can actually, after this prolonged period of time, we can actually see actually numbers of dysfunctional CD8 T cells that were similar to those that we can find in mice, in which intrahepatic CD8 T cell activation was very well proficient. Those CD8 T cells were highly proliferating, and if we did single cell RNA sequencing, we can actually very well clusterize those two conditions very well in this UMAP. So in the red condition, we can appreciate the HC prime cells, and in blue, the KC prime condition. Then we try to annotate those cells. As you can see here, our dysfunctional T cells were highly positive for PD-1 lactate and tox, which are prototypical marker for T cell exhaustion. And here, this orange cluster, because of the expression of Ki67, we annotate them as proliferating cells. This pink population here was very interesting because of the expression of TCF7, which encodes for TCF1, which is a transcription factor associated with stem-like properties. We annotate this population like dysfunctional T-stem-like. All the other one, because of the expression of the tissue resident marker 6R6, we annotate them as dysfunctional TRM1 and 2. I'm not going into details, but there's some further differences in the 1 and 2 TRM population. So we try to generate a score signature of T cell exhaustion that we can find by this publication in Nature Immunology a few years ago. And if we superimpose this dysfunctional signature to our, this exhausted signature to our dysfunctional cells, you can appreciate that it's not that high. I mean, it's kind of exhausted, but can you see the color scale is not that highly exhausted, meaning that it's kind of different from the canonical exhaustion. And if you look at the expression of the OX40 and 4-1BB co-receptor, we found that although 4-1BB was homogeneously expressed across all the different dysfunctional subsets, but importantly was not present in the other canonically exhausted T cells, the OX40 expression was specifically retained on the dysfunctional stem cell-like cells. So finally, we wanted to challenge these chronically infected mice with the monoclonal antibody for OX40 for MBB. And for doing that, we adopted transfer naive T cells. We wait 28 days, and then we challenge with 4-1BB or OX40 agonists, and the day after we evaluate the activation of T cells. As you can see here, 4-1BB was the only that showed the capacity to reinvigorate CD8 T cell, and you can find here by increased ALT elevation and the presence in this immunostaining of case-space 3. So our previous work showed that the hepatocyte-primed CD8 T cells poorly respond to PD-1 blockade, but they can be functionally rescued by interleukin-2 or IL-2. The problem is that IL-2 is not an ideal therapeutic agent because for its pleiotropy, it activates many different immune cells. It causes systemic toxicity and has a very short half-life. So the question here in this second part of the talk became, how can we deliver the benefits of the IL-2 while avoiding those limitations in terms of toxicity and pleiotropy and specificity? IL-2 has been known for decades as one of the most potent cytokines for boosting T-cell response, and I want to highlight that it was the very first immunotherapeutic. And back in the days, in the 80s, some patients with advanced metastatic cancer, melanoma, has been completely treated with a single interaction of IL-2. Of course, this excitement was short-lived because high dose of IL-2 caused vascular leak syndromes, severe systemic toxicity, which prevents the further development in clinical practice and clinical use. So to understand why that, briefly, I would like to look at the IL-2 receptor. The IL-2 receptor is composed of three subunits, the alpha, the beta, and the gamma, which the beta and gamma form the intermediate affinity receptor, and it is responsible for signaling, whereas the alpha chain, which is the high affinity receptor, which is also called CD25, does not signal itself, but increase the receptor affinity for IL-2 by about 100-fold. As a result, different immune cell populations respond very differently to IL-2 depending on which receptor subunits they express. For instance, regulatory T cells express high levels of the high affinity receptor, which makes them very, 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 very potently sensible to IL-2, and which leads actually immune tolerance rather than immune activation. One strategy to overcome IL-2 toxicity has been the development of IL-2 mutants over the days. So the idea is very simple. We should introduce a mutation in the region of the IL-2, which binds the alpha chain, so the CD25. And by reducing or abolishing this interaction, IL-2 can no longer efficiently engage with the high affinity receptor. As a result, the signaling is shifted towards cell expressing the intermediate affinity receptor, which is the beta and gamma, such as activated CD8 T cells, but of course also NK cells, which express very, very, very high level of the beta and gamma receptor. And take note of these NK cells can cause systemic toxicity if they are too much activated. This receptor bias, so the introduction of these IL-2 mutants, has greatly improved the therapeutic potential of IL-2, but still does not provide cell specificity. For that reason, and based on this rationale, in collaboration with this biotech based in the US, so the Asher Bio, we developed a CD8-targeted IL-2. And as a benchmark, we use an untargeted IL-2 mutant which represent the current generation of the IL-2 therapeutics that I just mentioned to you. So although these molecules avoid signaling through CD25, they still activate NK cells, such as, because NK cells express high level of the IL-2 receptor beta and gamma. So our strategy was a little bit different. So we further reduced the affinity of IL-2 for the beta and gamma receptor, making this our IL-2 cytokine basically inactive on its own. So then we fuse this mutant to an anti-CD8 antibody in a such way that when the antibody binds to its target, so a CD8 T cells, IL-2 is presenting insists on the same CD8 T cells. And so this optimal presentation restores the productive receptor engagement and signaling pathway downstream. So as a result of this strategy, IL-2 activity becomes largely restricted to CD8 T cells, minimizing the signaling to bystander immune population, such as Tregs, which drives unwanted immune suppression, or NK cells, which drives unwanted toxicity. So we first evaluated both molecules in wild-type mice to assess their safety profile. And I wanted to show here that we actually inject those compounds, the control IL-2 and the CD8 IL-2 in wild-type mice, and we did not observe any sign of systemic toxicity as body weight loss, neither any sign of liver-specific toxicity, as you can see appreciated here by no increase of ALT elevation, and the histology of the liver were absolutely normal. Importantly, both molecules, the control and the CD8 IL-2, both were able to expand in the spleen and in the liver CD8 T cells. But I want you to appreciate that the CD8 T cells rescued by the CD8 IL-2 were much more potent to induce the expansion of CD8 T cells compared to the control IL-2 molecules. However, when we look more closely at the responding cell population, an important difference emerged. So the untargeted IL-2 mutant still strongly signal to the other IL-2 receptor expressing cells like group 1 analysis, which comprises also NK cells. As you can see here, we have this is a log scale, so we have a huge expansion of group 1 analysis in the spleen and the liver, and also of Tregs. But if you look at the capacity of CD8 IL-2 to expand those populations, we actually found that the numbers of this population were absolutely normal if you compare them to the control untreated condition. So this confirmed that the cis targeting approach not only enhanced CD8 T cell activity, but also greatly improved the cellular selectivity. So the next question was whether this increased selectivity would translate into better antiviral efficacy and activity in our HBV model. In order to answer this question, we take our HBV transgenic mouse model, and we adoptively transfer those recipient mouse with 10 to the 5th CD8 naive T cells, and the day after we challenge those mouse with the control, with control IL-2 or CD8 IL-2, and we analyze the fate and the antiviral differentiation of these T cells over time. CD8 IL-2 induces stronger expansion of HBV-specific CD8 T cells and restore their effector function, as shown here by absolute number and by increased interferon gamma positive T cells that we can collect from the liver of those mice. Most importantly, this translating into the marked reduction in viral DNA, in systemic viral DNA in the viremia, demonstrating superior antiviral activity compared with the untargeted IL-2 mutant. As expected, this enhanced immune response was associated with an transient increase in liver inflammation, reflected by serum ALT increase, which again indicate immune-mediated clearance of infected hepatocytes. So as a conclusion, as a summary of this second part, I hope to convince you that conventional IL-2 mutants still activate multiple immune cell population, creating a cytokine sink limiting the amount of IL-2 available for antigen-specific CD8 T cells. By contrast, CD8 T cells, CD8 IL-2, deliver the cytokine directly to antigen-specific CD8 T cells to cis targeting, and this selectiveness restores the antiviral function while minimizing the off-target immune activation. And these results demonstrate as a proof of concept that cell-specific cytokine delivery can substantially improve both the efficacy and selectivity of the IL-2 based immune therapy. So this is great. We have collaborated with Asher Biotech, but then while these results were encouraging, we wanted to go further and develop our own IL-2 platform. So the idea, and this last part of the presentation is unpublished work. Our idea was to use the lipid nanoparticle platform, similar to those used in COVID-19 vaccine, to deliver IL-2 messenger RNA directly to the liver. So in order to achieve a hepatocyte-specific expression, we incorporated in the sequence of the messenger RNA microRNA sequencing. So these sequences are recognized by microRNAs expressed in hematopoietic cells, meaning immune cells in the circulation, but not only, and endothelial cells. How does it work? So the recognition of this mRNA sequence lead to the degradation of the transcript and preventing off-target expression of the IL-2. In contrast, a parasite does not express those microRNAs, and therefore, when the lipid nanoparticle deliver its cargo within the cell, they efficiently translate into IL-2, creating a local source of the cytokine within the liver and exposing only intrahepatic T-cell, CD8 T-cells to the selective IL-2 gradients. So first of all, before starting and testing efficacy, we wanted to confirm the target engagement. So following the administration of IL-2, we did some confocal imagings and the parasite express IL-2, as shown here. But then we moved forward and we wanted to evaluate the safety profile of this platform in wild-type mice. So we actually inject 0.3 milligram per kilo of the IL-2 messenger RNA lipid nanoparticles in wild-type mice, and we analyzed different parameters. We found no evidence of systemic or liver toxicity, as we did not find any body weight increase, we did not find any ALT elevation, and the micrograph of the liver histology were basically healthy. At the same time, we observed a very modest expansion of IL-2 receptor expressing immune cells, specifically into the liver, whereas the spleen remained largely unaffected. So this finding indicates that the IL-2 expression is largely confined to the liver, where it induces local immune activation without causing systemic toxicity. So to further optimize this platform, we collaborate with Professor Norbert Party at UPenn, University of Pennsylvania, which is a pioneer in messenger RNA lipid nanoparticles technology. And together we generate 3 different lipid nanoparticle formulation. We changed different lipids, ionizable lipids, and not only, and all carrying the same IL-2 messenger RNA sequence. And we compare their performance in our HBV transgenic mouse model. So we adopted transfer once again our CD8 T cells, and the day after we injected 0.05 milligram per kilo of the 3 different compounds, and we analyzed 5 days later. So all the formulation were well tolerated. We did not observe any important body weight loss. All mice survive, and however, they differ substantially into biological activity, as you can see here by systemic ALT elevation. One formulation in particular, the formulation 3, induced a much stronger activation in immune responses, as mirrored here by increased ALT elevation, which might be consistent with more efficient immune-mediated clearance of infected hepatocytes. We next compared the antiviral activity of the formulation, and as you can see here, all the 3 formulation promoted the expansion of HBV-specific CD8 T cells. Here, it's noted that absolute numbers of CD8 T cells collected from the liver. But importantly, again, the third formulation performed better. We also took these CD8 T cells and we stimulated in vitro with the coconut peptide, and the lipid nanoparticle formulation number 3 showed the greater potential to promote interferon gamma production in antigen-specific CD8 T cells, as well as granzyme B, which is very important for antiviral immune function. Finally, we asked whether the improved CD8 T cell function also translated into viral clearance. As you can see here, the HBV core antigen staining in the liver compared to the PBS control, all the 3 formulations reduced the amount of the viral antigen. However, once again, the formulation number 3 was clearly the most effective, with an almost complete loss of the HBV core staining, indicating near complete viral clearance. So these histological data are consistent with the functional and virological results that we observed in the previous experiments and in the previous data. So in summary, we developed an hepatocyte-targeted IL-2 messenger RNA platform based on lipid nanoparticles. The messenger RNA is selectively translated by hepatocytes, creating a local source of IL-2 within the liver, while preventing off-target expression in other cell type. And this local IL-2 is able to rescue the function of dysfunctional HBV-specific CD8 T-cells, which in turn restore the cytokine production and the cytotoxic activity. And finally, this results in efficient elimination of infected hepatocyte and the control of the viral replication. So before I finish, I'd like to acknowledge all the people who contributed to the work. First of all the members of the Iannacone Lab, whose dedication made this study possible. I'd like to acknowledge also our collaborators, in particular Professor Luca Guidotti, Professor Norbert Parti, UPenn, as well as Ivan and Kelly from the Asher Biotech, who were instrumental for the L2 engineering and the study, and all the funding agency for supporting our research. And thank you very much for your attention. I'd be more than happy to take any question. Thank you very much.
Thank you so much. That was very insightful. I want to remind everybody that if you have any questions, you can put that in the little Q&A box down at the bottom, and then we will take your questions. So to start off, I have a question about if there are any strategies that are applicable for direct delivery into CD8 T cells.
So this is a very important question. Thanks for answering for making it. This is a very relevant question because we started with this approach, designing lipid nanoparticles for targeting the hepatocytes, which is very convenient because the liver is known to be to have a very important tropism for lipid nanoparticles. So targeting hepatocytes is relatively easy because those are lipids, and the systemic circulation, they complex with a very complex protein crown, which included also apolipoprotein or lipoprotein, and hepatocytes are very rich of lipoprotein receptor. So lipid nanoparticles goes there in the hepatocyte very efficiently. Of course, this is very good for us because we want to rescue intrahepatic T-cell immunity. But of course, the idea would also to deliver messenger RNA, antisense oligonucleotides. I just mentioned few compounds that we are trying to test, either to promote the expression of a given gene or to knock down on the expression of a break gene, for instance, which is responsible for the differentiation of this dysfunctional phenotype. So for CD8 T cells, it's much more complicated because they are less well transduced and targeted compared to hepatocytes. So now the idea would be to use, first of all, to change the composition, making those lipid nanoparticles less tropic for the hepatocytes, but also to functionalize these lipid nanoparticles with antibodies targeting antigens expressed of CD8 T cells. I just present you the work of CD8. So CD8 might be a target, CD3 or CD5 or CD7, or maybe TIN3. I don't know. I just mentioning a few receptor that might be targeted. So this is something that we are actively exploring now in the lab. It's challenging, but eventually it might be possible to make it. Why not? This is very important as well.
Thank you so much. Related to that, do you think the strategy could be applied beyond hepatitis B, such as other viral infections?
Absolutely. I mean, you mean the lipid nanoparticle platform? So this one, yes. I mean, might be interesting for other viral infections in the liver, because the idea is to produce immunostimulant cytokines like IL-2 specifically in the liver. So it might be, of course, for sure, the case also for other chronic infection, but also it might be also very interesting in cancer condition, because for that metastatic cancer in the liver has a very poor prognosis because of the liver tolerogenic environment. Either the primary tumor, the hepatocellular carcinoma, but liver mets are very difficult to treat because the liver is, for some mechanism that we are studying, is can impose this dysfunction not only in chronic HBV infection. For sake of time today, I did not show you those data, but we know in our preliminary work that the liver-imposed dysfunction is shared across other chronic infection in humans and in cancer. So the idea would be to use this specific delivery of, once again, messenger RNA into the panocytes to create an immunostimulant environment in the liver, also for other chronic settings like cancer and possibly other chronic infections. So that's a very relevant question, and I would say yes, definitely.
Thank you so much. I have another question. If this LNP mRNA was to be developed as a treatment, how long would you expect a patient to receive this treatment for? The person noticed that cccDNA was not addressed, so this treatment would only suppress and imagine infection would return at some point. Have you assessed that in mice? Do they make it? And they can analyze it if you do an alkaline lysis.
So you're asking whether the cccDNA was successfully eliminated, eradicated in the recipient mice. So this is another very important question. This HBV transgenic mouse model, the cccDNA, which for the one in the audience are not familiar, which the HBV pathogenesis is this part of this genome, the genome of the HBV, which persists in the cells and we in the parasites, and we need to eradicate as well this reservoir of the viral genome to get the sterilization of it. So in this transgenic mouse model, the cccDNA is very difficult to see and to analyze. Now we are trying to, with other collaborators, to improve the expression of the HBV genome in order that can be actually be detectable by Stauder blot, for instance, so other techniques. Concerning the question on the translatability of these lipid nanoparticles in the clinic, of course, this is something that has been tested in preclinical models, but the idea is that we are using lipid nanoparticles platform that were worldwide used for the pandemic, and nowadays they are more and more used also in cancer clinical setting. So the idea would be to use this platform, which is already regulated by the agency, the agency. So the idea is to make all this platform quickly, in a quicker way, ready to be tested in humans as well. How far are we from that? I don't know. Really don't know to answer this question.
No, that's fair. Thank you so much. Something kind of related, for using agonists of the co-stimulatory receptor for 1BB, what do you think is the biggest hurdle to translating the strategy into the clinic?
That's another very important question, because for 1BB is known in the preclinical practice and in clinic that is a very potent immune activator, and clinicians have observed very important liver toxicity for 1BB. So this is something that we have to be carefully explore. In our model, we did not observe any systemic toxicity or liver-specific toxicity with the 4-1BB agonism, but possibly because we use very little amounts of 4-1BB in one single dose. So we know that if we inject it multiple times, this agonist, we actually might eventually ended up to observe liver toxicity. So for this reason, now we are collaborating with another lab to develop other 4-1BB chimeric antibodies with other strategy in order to be more selective for dysfunctional T cells in order to selectively deliver the 4-1BB T cells, because it seems that NK cells are the one that once targeted by 4-1BB are responsible for this liver-specific toxicity. So the idea would be to create this chimeric 4-1BB agonist linked with the other immunostimulatory modulants, so that we can go down with the dose while preserving maximal immunotherapeutic effects.
Cool. Thank you. We have another question. Do you expect a differential response to therapy between acute and chronic HBV infections?
That's a very fair point. Uh, I don't know. What I can say is that chronic and acute infection are very different because of different phases of the infection. So we have the very first phase, because I mentioned in the introduction that HBV is non-cytopathic, is invisible to the innate immune system detection machinery. So the very phase is immune tolerant, which means that the virus replicates a lot, but the body and the host does not really realize that the viral infection is going on. So there's no inflammation. So CD8 T cells that get primed into the liver, which is tolerogenic, and in the total absence of inflammation, which is very importantly, you know that inflammation is driving the expression of PD-L1. So a CD8 T cells can express a lot of PD-1, but the immunotolerant phase, there will be no PD-L1 because there's no inflammation. So this strategy may have a beneficial effect. In the chronic phase, it's more difficult to say, because you have this over time, you have this low inflammatory response, maybe subclinical, but over time, eventually, the accumulation of this low cycle inflammation will eventually develop cirrhosis and hepatocellular carcinoma. So in this chronic phase, it's hard to see, but I would definitely expect some differences. What I can say is that we have tried with PBNC, they're not intrahepatic cells, but with PBNC of chronically infected patients. And for maybe it works better than PD-1 and OX40. So, yeah, definitely.
Thank you so much. We have another technical question. Will IFN or unregulated PD-1 be a concern during chronic antigen exposure?
Can you say it again? Sorry, whether type 1 interferon
Yeah, I think so. Will it be a
is it
concern during chronic antigen exposure?
can you say it again? Whether
The
the
IFNR, unregulated PD-1.
um, uh, I don't know. To be fair, I don't know. Possibly yes, but I don't know how to answer this question. Whether the PD-1, the regulation might be a problem. Possibly yes.
That's fair. Thank you. All right, we have one last question to close out the Q&A. Unless anybody else has any other questions, please put in the box. So we have, what do you think are the limitations with the HBV mouse models that you mentioned in the beginning of the talk, if there are any?
Absolutely. Of course, there are a lot of, I mean, there are limitations. These are preclinical models. And as I was mentioning in the introduction, HBV does not infect mice. So that's why we ended up with transgenic mouse model. First of all, the first very important limitation is that it's not a model of infection, because maybe I can go back to the very first slide just to show the limitation here. Yeah, so here the parasites express high level of HBV particles, which are infections, because if we took these particles, the serum of these mice, and we transfer in chimps, for instance, those chimps get infected. So they are highly infectious, and this is why we host these mice in our BCL3 facility. But the problem is that because the mouse is not targeted cannot be infected by viral, HBV viral particles, they cannot go back and recycle back and infect new parasites. So that's one limitation. So this mouse model can be used for studying the antiviral activity, but not the infection phase. Second, since it's a transgene here, it's kind of the mouse is immune tolerant, so that's why we have to adoptively transfer CD8 T cells. because it's different. It's difficult to block the tolerance. We know that CD4 T cells are not involved in controlling the peripheral tolerance is not involved in controlling this. So this model is quite fair. But of course, this is a transgene, so this has to be taken into consideration when translating this finding to the humans as well. So those are the first two bigger limitations that I can see. But of course, this mouse model has been used in the past decades to develop all the antiviral that we are currently using in the clinic, like the capsid modulators or other antivirals that we use have been also tested and developed in these synthetic mice.
Awesome. Thank you so much. That was absolutely wonderful, very informative. Yes. And I'm just making sure there are no more questions. Yes, that is the end of all our questions. And thank you so much, everybody, for attending. And thank you, Dr. Francesco, for your time and that wonderful, wonderful talk.
It was a pleasure. Thank you very much for the invitation. Bye.
Yes. Bye.